Thanks for joining us in another fascinating journey through the world of science here on the Sands Channel podcast.
Great to be here.
If you're finding these deep dives into scientific literature intriguing, please do take a moment to like this and subscribe. It genuinely helps us bring you more of these explorations.
It really does.
So today we're tackling a seemingly simple concept with, well, surprisingly complex implications. Temperature. We'll be diving into the compelling link between body temperature and how different organisms age and, how long they live.
From tiny insects all the way up to us mammals.
Exactly. Uncovering some unexpected connections and questions that are still being actively researched. It's almost intuitive, isn't it, to think that the hotter things run, the faster they wear out.
Yeah, that's been a long-held idea in biology. Faster metabolism, higher temperature, faster aging. It seems logical.
But, as we're about to see, the reality is, well, far more intricate. Our mission in this deep dive is to explore the scientific evidence for how temperature influences lifespan and delve into some of the fascinating biological mechanisms that might be at play, drawing directly from the research.
It's actually quite remarkable to think that something as fundamental as temperature can exert such a profound influence on, well, the very fabric of life.
Right.
The initial thinking, stemming from basic physics thermodynamics, suggested higher temperatures, more energetic molecules, increased motion. It could inherently lead to more cellular damage, a faster rate of aging.
Okay, so let's start by looking at a broad trend. One that emerges when we examine this relationship across many different types of organisms. It appears that, regardless of whether an organism regulates its own body temperature or not, lower body temperatures are often associated with longer lifespans.
Yeah, that pattern holds up surprisingly well. And it's not just theoretical. We see it in the wild and in, controlled lab settings too.
So where did the first hints of this come from?
Well, some of the earliest and I think really convincing evidence came from studies on fruit flies. Drosophila melanogaster.
Ah, the classic model organism.
Exactly. Researchers found these tiny insects lived about twice as long when kept at a cooler 21 degrees Celsius compared to a warmer 27 degrees Celsius.
Wow, twice as long. That's significant.
It really established a strong initial link. Lower temperature, longer life, at least in an ectotherm, an animal that relies on external heat.
And this wasn't just a one-off finding in flies, was it?
No, not at all. We've seen similar patterns in other invertebrates, things like the small crustacean Daphnia magna.
Okay.
And also in various fish species, like those South American Cenalebius fish.
And what's striking is that even quite minor but sustained shifts in temperature could make a difference.
Absolutely. Yeah. Even relatively small, consistent drops in temperature can act as this powerful lever, kind of extending lifespan, often by quite significant margins in these cold-blooded creatures.
So focusing on invertebrates then, their dependence on outside temperature must make them easier to study in this context.
Precisely. It's much easier for researchers to manipulate this variable, which gives us some really valuable insights. Take C. elegans, the tiny worm used so much in aging research.
Right.
A mere 5 degrees Celsius drop in its environment. That resulted in an astonishing 75% increase in its lifespan.
75%? Just from a 5-degree shift. That's incredible.
It really is. And this pattern seems pretty consistent in many insect studies, too. For example, certain wasp species, a similar 5-degree drop boosted their median lifespan by over 70%.
It really points to a fundamental connection there, in ectotherms at least.
And what's particularly interesting is how lower temperatures in these invertebrates seem to link up with a generally slower pace of life.
Slower pace? How so?
Well, you often see slower rates of development, for instance. And this actually lends support to that older idea, the rate of living hypothesis.
Ah, the idea that if you live slower, you live longer. Less wear and tear.
Kind of, yeah. Slower metabolic rate, often tied to lower temperature, could simply mean it takes longer for the organism to accumulate the, damage associated with living.
That makes a certain intuitive sense, like running an engine slower so it lasts longer. And you mentioned some extreme examples.
Oh, yeah, absolutely fascinating ones. These incredibly long-lived organisms in freezing environments, like certain Antarctic sponges.
Sponges? Living for how long?
Well, things like Senecera antarctica and Scalimaster jubini, they grow incredibly slowly in those frigid waters. And they're thought to potentially live for centuries, maybe even millennia.
Millennia, wow. That really highlights how dramatically temperature can scale the whole time scale of life.
It does. And if we connect this to the bigger picture, we see similar trends in vertebrate ectotherms too, fish, amphibians.
Okay, so moving up the chain a bit.
Evidence from wild populations suggests those living in colder places tend to live longer. Think about fish species, where populations in colder northern waters often grow slower, mature later, and ultimately live longer than their cousins in warmer southern waters.
So it's not just a lab thing, it's playing out in nature.
Exactly. And we have compelling lab studies, too. Those short-lived Centolebius fish, again dropping the water temperature, significantly extended their lifespan.
Right.
And more recent work on other short-lived fish, like Nothobranchius furseri and Nothobranchius rotovi, has pretty much confirmed this.
What about amphibians, frogs, salamanders?
Well, the data there is mostly from field studies, which makes it a bit trickier to isolate temperature specifically.
Sure, lots of other factors in the wild.
Right. But the general observations do align. Greater longevity seems linked to colder environments, or perhaps more strongly, higher altitudes where temperatures are generally lower.
Okay, so a pretty consistent picture in cold-blooded vertebrates as well. Now let's shift gears to homeotherms. Animals like us, mammals, birds, who keep a stable internal temperature. Here, the relationship seems to get, well, a bit more complicated. We have a much narrower range of internal temperatures.
Precisely. That straightforward inverse relationship we often see in ectotherms. It's not always so clear-cut in mammals.
But there are still hints, right? You mentioned some intriguing cases.
Oh, definitely. Take the Ames dwarf mouse. It's famous for its extended lifespan.
Right.
And these mice also happen to have a lower body temperature. It's potentially linked to deficiencies in certain hormones involved in growth and metabolism, like TSH and growth hormone.
Interesting. And you also mentioned GHR knockout mice. Similar effects?
Yeah, they also live longer and show lower insulin and IGF-1 levels, which kind of mirrors what we see with caloric restriction, another lifespan extender we should talk about.
Okay, we'll get to that. But is lower body temperature always required for these long-lived mutant mice?
That's a crucial point. No, it isn't. Life extension can happen through various biological pathways. It's not a universal rule, even in these specific mouse models.
But there was a key study that provided more direct causal evidence in mammals.
Yes, a really significant one involving genetically modified mice, the HTRT-UCP2 mice. They managed to engineer a very modest reduction in their core body temperature.
How modest.
Only around 0.3 degrees Celsius. Tiny.
And the effect?
A notable increase in median lifespan, something like 12 to 20%.
Wow, that small a drop, that big an effect, that does suggest a causal link in mammals.
It really does. But then science is never simple, is it?
Aha, rarely.
There was another study in a different mouse strain, MF1 mice, that didn't find a direct correlation between an individual mouse's body temp and how long it lived.
Oh, what did they find instead?
They suggested maybe metabolic intensity was more positively correlated with lifespan, though it's worth noting they didn't report much variation in body temperature among their mice, which makes it harder to draw strong conclusions about temperature itself.
So conflicting results highlight the complexity in homeotherms.
Exactly. And then you throw in things like gender differences.
No.
Well, in some mouse strains, females tend to have slightly higher body temperatures than males, and they also tend to live shorter lives.
Suggesting maybe a complex interplay, perhaps involving sex hormones affecting both temperature and aging.
It's certainly possible. It's all interconnected.
And the human example really throws a wrench in the simple lower is better idea, doesn't it?
It absolutely does. Women, on average, tend to have a slightly higher core body temperature than men.
Yet women generally live longer than men.
Exactly. So that simple equation, lower temperature equals longer life, clearly doesn't hold universally for warm-blooded animals like us, where temperature is so tightly controlled.
But there was that study, the Baltimore Longitudinal Study of Aging.
Yes. That study did identify lower body temperature as a biomarker that correlated with higher survival rates, specifically in healthy older men.
Okay, so even in humans, while it's not the whole story by any means, temperature might still be playing some role in the aging process.
It certainly seems plausible, yeah. It's likely one factor among many.
Let's bring in caloric restriction now, CR. It's probably the most well-known intervention for extending lifespan in many organisms. And interestingly, CR often leads to a drop in core body temperature. How does that fit in?
It's a very relevant piece of the puzzle. In rodents, CR typically causes a drop of about 1 to 1.5 degrees Celsius in core body temperature.
Okay, quite a bit more than those genetically modified mice.
Yes. And this led to the hypothesis that maybe, just maybe, some of the life-extending magic of CR is actually mediated through this drop in temperature.
But you mentioned earlier that CR and just lowering the ambient temperature seem to affect mortality differently, at least in fruit flies.
Exactly. That research suggested they might be working through different biological pathways. CR in flies seem to mostly delay the onset of age-related mortality.
Pushing back the start of the decline.
Right. Whereas lower environmental temperatures seem to actually reduce the slope of the mortality curve.
Meaning it slowed down the rate of aging itself.
That's one interpretation, yes. It suggests temperature might be affecting the fundamental rate of aging, while CR might be more about delaying age-related diseases or dysfunction. It's subtle, but potentially important.
And is the temperature drop with CR always consistent? Does more drop equal more lifespan extension?
That's where it gets tricky again. It seems the relationship isn't always straightforward. You pointed out that in some mouse strains, surprisingly, a greater drop in body temperature under CR actually predicted less lifespan extension.
Really? That seems counterintuitive if temperature reduction is the key benefit.
It does. It strongly suggests that the benefits of CR aren't solely dependent on lowering temperature. There must be broader cellular and metabolic adaptations involved, maybe linked to how the body handles energy, fat storage, things like that.
But we do see temperature drops with CR in other animals.
Oh, yes. Studies on Rhesus monkeys under long-term CR showed a decrease in body temperature, which supports the idea of energy conservation being part of CR's effects.
And in humans.
Human studies on people practicing long-term CR have also shown a modest reduction in core body temperature, potentially linked to changes in thyroid hormone levels.
Okay. So CR often lowers temperature, but how much that specific temperature drop contributes to the lifespan benefit seems variable, and maybe not the whole story.
Exactly. Which brings us nicely to the question of the actual biological mechanisms. Why does lower temperature often correlate with longer life? It's probably more than just slowing things down, right?
It seems that way.
Absolutely. While that thermodynamic idea, lower temps, slower reactions, less molecular damage, is a logical starting point.
Like atoms jiggling around less.
Sort of, yeah. But the biology seems way more intricate. For instance, those housefly studies showed longer life at lower temps, but also less activity. So there are trade-offs.
Right, and then there's that other concept, hormesis.
Yes, hormesis. The idea that mild stress, maybe even a short burst of slightly higher temperature in some cases, can actually trigger protective mechanisms in cells.
Making them more resilient in the long run.
Potentially, yes. It's like exercising a muscle to make it stronger. A little stress can sometimes be beneficial.
So it's not always about minimizing stress or temperature.
Okay.
And the specific ways lower core body temp extends life in those mice. Still being worked out.
Still under active investigation, definitely. Possibilities include things like improved energy efficiency at the cellular level, maybe reduced production of harmful reactive oxygen species, oxidative stress, or less DNA damage accumulation.
Things that are hallmarks of aging.
Exactly. And the fact that CR's effects on lifespan aren't perfectly tied to temperature reduction really hammers home that other biological factors are crucial.
So what are these other factors? You mentioned active biological mechanisms.
This is where recent research gets really exciting, especially in simpler organisms like C. elegans and fruit flies. It's revealing active biological pathways. It's not just passive physics.
Active pathways, like what?
Well, they found things like temperature-sensitive sensory channels on cells. Think of them like tiny thermometers.
On the cells themselves.
Yeah, TRP channels in C. elegans, for example. When these sense a drop in temperature, they trigger a whole cascade of signals inside the cell, involving molecules like calcium, PKC, SGK1,
Okay.
and ultimately influencing a key longevity regulator called DAF16, or FOXO in mammals.
FOXO pathways are heavily linked to aging and stress resistance.
Precisely. So the worm isn't just passively slowing down, it's actively sensing the cold and switching on longevity programs. Similar findings are emerging in fruit flies, too. Physiological adaptations to cold seem important.
That's a huge shift from just thinking about slower chemical reactions. It's a programmed response.
It really is. It challenges that purely passive thermodynamic view. And it opens up the possibility of, neuroendocrine mechanisms playing a role, too. Hormones mediating the response.
Hormones involved in growth and development?
Yes. Temperature clearly affects those in ectotherms. The GH-IGF system in fish seems involved. Even some hints in humans about exercise temperature and growth hormone. It's a complex network.
Any other proposed mechanisms?
Well, there are other hypotheses floating around. Maybe lower temps boost pathways that suppress autoimmunity or increase resistance to environmental stresses generally.
Okay.
And then there's brown adipose tissue, BET, The good fat that generates heat.
Right, through uncoupling proteins, UCPs.
Exactly. Bead activity, mediated by UCPs, might reduce harmful ROS production, potentially linking to longevity, though BD activity does tend to decline as we age.
Interesting link there. And didn't you mention cold exposure in mice correlating with less body fat and maybe fewer tumors?
Yeah, that's another observation that adds to the complexity. It really suggests the whole relationship is multifaceted. It involves energy balance, stress responses, possibly immune function, way beyond just simple metabolic rate.
Okay, so speaking of metabolic rate, how does this all look when we compare across different species of homeotherms like mammals? Does the rate of living idea hold up there?
This is where that simple theory really breaks down. Well, you have some homeotherms, like hibernators, that undergo huge drops in body temp during torpor, and that is associated with increased survival and slower life histories. Bats that hibernate longer tend to live longer. Smaller hibernating mammals tend to have longer maximum lifespans for their size.
So that fits the pattern.
It does.
Yeah.
But look more broadly across all homeotherms. The picture gets messy. Take birds.
Okay.
Birds generally run higher body temperatures than mammals of similar size, but they often live much longer.
Oh, right. That completely contradicts the simple idea.
Totally. And then you have marsupials. They tend to have lower body temps compared to placental mammals like us, but generally have shorter maximum lifespans.
So higher temp, longer life in birds, lower temp, shorter life in marsupials? It's all over the place.
Pretty much. There was a big study looking across many mammal species. It found a link between body temp and basal metabolic rate, which makes sense.
Hotter bodies burn more energy at rest.
Right. But crucially, they found no correlation between that basal metabolic rate and longevity across species.
Not at all.
Nope. And only a weak negative correlation between body temperature itself and longevity across mammals.
So being slightly cooler might have a small benefit on average across mammals, but metabolic rate itself isn't the key predictor of lifespan between species?
Exactly. Things like ecological factors, evolutionary history, body size, and maybe how temperature influences the timing of life events seem much more important than just the overall metabolic speed when comparing species.
Okay, this complexity really sets the stage for our final example. The naked mole rat.
Ah, yes. The superstar of weirdly long-lived rodents.
Right. Longest-lived rodent, incredibly cancer-resistant, and really unusual temperature regulation for a mammal.
Absolutely. They're fascinating because, unlike most mammals, they're basically poikilothermic. Their body temp fluctuates much more with the environment.
More like a reptile in that sense.
Kind of, yeah. And they maintain a low metabolic rate and a low body temperature compared to other mammals their size.
Which immediately makes you wonder, is that low temperature part of their longevity secret?
It's the million-dollar question, isn't it? Their typical body temp is around 32 degrees Celsius.
Compared to maybe 37 for a mouse, that's a big difference.
It is. So it seems like a prime candidate to support the lower temp, longer life idea.
There's always a but.
There is. Pregnant females have higher body temperatures. And the queens, the breeding females who live the longest, don't seem to have shorter lifespans because of it.
That complicates the picture within the species itself.
It does. Now, researchers have tried to estimate the impact. They've extrapolated from studies on other animals.
It's like asking, what if a mouse lived at 32 degrees?
Exactly. And those estimates suggest mice kept at cool might live considerably longer, maybe 4.5 to 7 years instead of 2 or 3.
Wow.
And conversely, if a naked mole rat lived at a mouse's temperature, its lifespan might be shorter, maybe 20, 25 years instead of 30 plus call.
But these are just extrapolations, right? Huge caveats apply.
Huge caveats.
Yeah.
Extrapolating across species is always risky. So the consensus is that the low body temperature likely contributes to the naked mole rat's longevity.
But it's definitely not the whole explanation.
Not by a long shot. Other unique adaptations are clearly crucial. Interestingly, other mole rat species also tend to run cooler than other mammals. And the naked mole rat is the coolest and longest lived of the bunch.
Okay, so there's a pattern within that group.
But then you look elsewhere. The eastern gray squirrel, relatively long-lived for a rodent, but has a higher body temp.
Right, another counterexample.
And even the longest-lived mammal, the bowhead whale, has a relatively low body temp for its massive size. So maybe it contributes a bit there too, but again, likely a small part of a much bigger picture.
So wrapping this up, it seems temperature undeniably influences longevity across the board, from flies to whales.
Clearly a significant factor, yes.
But the how and why are far more complex and varied than that simple rate of living or thermodynamic idea suggested.
Absolutely. The mechanisms involve active biological responses, neuroendocrine signaling, metabolic adjustments. It's intricate.
The emerging understanding of these active responses, those sensing pathways, seems particularly exciting.
Definitely. It opens up whole new avenues for thinking about aging.
So, thinking ahead. Considering all these diverse ways temperature interacts with aging across species, what do you think might be the most surprising direction for future research, especially when it comes to maybe influencing longevity in humans?
That's the really provocative question, isn't it? Maybe it's not about drastic temperature changes, but finding ways to subtly modulate our internal temperature set points, or perhaps enhancing those cellular resilience pathways that respond to temperature cues.
Tapping into those active mechanisms you mentioned.
Exactly. Understanding how C. elegans, or fruit flies, actively manage longevity in response to cold might hold clues, maybe not directly applicable, but pointing towards conserved pathways we could potentially influence for healthier aging in humans. It seems like we've only just started to unravel this really fundamental relationship.
A lot more to explore there. Well, this has been incredibly insightful.
Always fascinating to discuss.
As always, don't forget to like the video and subscribe if you haven't joined us yet.